So the disease state that, to extend the disease states, this is heart failure, which has been sort of a focus of my life for the last, well, since I was in medical school, I guess. And as you can see here in the US, and the numbers are exactly the same in the European Union, just multiply by 1.5, the incidence is increasing, especially with the aging population, and the prognosis for class 3, class 4 heart failure continues to be quite poor, sort of paralleling some very advanced cancers. One of the things that's been interesting in patients with heart failure is that you basically have the sort of a mixture of cells that are either non-diseased, dysfunctional ones, or permanently lost ones. And over the last 5-6 years, we've had a lot of work on cell therapy, which focus on replacing the permanently lost myocytes. Our work for now about 15 years has really been to target the dysfunctional cardiac myocytes. And that work really comes from pharmacological therapy for heart failure, which has really consisted of trying to target the sodium-potassium ATPase pump with digitalis, trying to go after beta receptor signaling and phosphorylating different sites within the cardiac myocytes, and more recently on activating the sort of the end effector of cardiac force development, and that's the myosin. So our work really is focused on calcium cycling, and obviously I'm not going to belabor the points about the issues of how to target different areas of the calcium cycling, but in cardiac myocytes, the SR calcium ATPase pump, circuit 2A, plays a central role in removing calcium during relaxation and storing it into the sarcoplasmic reticulum. And circuit 2A is downregulated and dysfunctional in failing hearts due to post-translational modifications. And that has been really the sort of target that we've been going after for many years. In addition, ryanodine receptors, which are also dysfunctional in terms of heart failure, have also been targeted with other therapies that try to stabilize the ryanodine receptor. So over the last number of years, we've taken a sort of a gene therapy approach because mainly pharmacological therapy failed when we were trying to target the SR calcium ATPase pump. And as Ken mentioned, we really had to sort of go after choice of vectors. For the cardiac tissue, we had to sort of work on modes of delivery. We had to basically address directly the immune response until we finally got to clinical trials. Now, in terms of the AAV vectors, as Ken mentioned, they're quite safe now. For heart failure, they have the characteristics of giving you a long-term expression, which is obviously important. There's a certain amount of cardiac specificity in terms of some of the AAV vectors, serotypes, and of course, there's a minimal immune response, especially at low doses, which is important when you're trying to treat a patient long-term. There are now hundreds as AAV serotypes, and I'm just showing you this slide that I borrowed from Jude Samulski, where you have different AAV serotypes targeting different organs. So, for example, 1, 6, and 9 are specific for muscle and heart tissue, 8 for the liver, 4 and 5 for the retina, and so on. So clearly, the serotypes of AAV vectors give you this added advantage that you can basically go after different organs by choosing different serotypes. Because this is a cardiac meeting, I just thought this would be interesting to show. This is an AV9 injection of a tail vein in a rat, and we're looking at the cross-section of the myocardium, and you can clearly see that the AV9 is infecting very well most of the cardiomyocytes, but look at the vessel here. So the vessel wall here and here are not infected at all. So clearly you can see that differential tropism of a vector within one slide. You know, all the myocytes are infected, but none of the vessel wall is infected. The other issue has come up in the last few years in cardiac gene therapy is that is AAV9 the best vector to use in because it has so much tropism. In fact, AAV9 is great for mice and rats, but if you look at large animals, which we've done, 1, 6, and 9, when delivered locally, have absolutely no differences. So yes, AAV9 is greatly tropic in rodent models, but in terms of its tropism or its efficacy in large animals and presumably humans, basically 1, 6, or 9 have very similar profiles. One of the things that is important to understand for AAV vectors is unlike adenovirus or other types of vectors, the expression really takes 2 to 3 weeks before it comes on. So here's an expression in mice that we were able to follow by luminometry. And you can see that the expression starts between 2 to 3 weeks. The nice thing, just like with any pharmacological treatment, is the more vector you have, the faster the expression and the higher the expression. So clearly you can dial up the dose and increase. Now, a number of years ago, we started out our program using AAV serotype 1, where we used a CMV promoter driving human SERCA2A. And this was a clinical grade vector that we produced. And just to summarize 10 years of work, basically preclinical work, we showed that SERCA2 gene transfer in the cardiovascular system increased contractility, so it restored the failing heart to more normal contraction. And so for a very cardiac myocyte specific crowd, it decreased ventricular arrhythmias, it improved energetic profile. There were a number of things that it did in terms of decreasing cardiac hypertrophy, and on extracellular, extracardiac cells such as endothelial cells and smooth muscle cells, it had bystander effects that were beneficial. And when we compare it to pharmacological therapy, which has failed for treatment of heart failure, so again, this is really one of the impetus of using a gene therapy approach, we're able to show that compared to the usual cyclic AMP dependent pharmacological increase in muscle function, we had improved energetics by targeting specifically SERCA2A, decreased arrhythmias, whereas pharmacological therapies worsen arrhythmias, and no change in cyclic AMP, which can be detrimental in patients with heart failure. And this led to basically a survival profile in multiple animal models of heart failure, where you had a significant improvement of survival in animals treated with SERCA2A. And again, this is in dire contrast to the ones that were treated with a cyclic AMP modulator, dobutamine. And I think this really illustrates one of the beauties of this targeted approach. The red curve and the blue curve, so the SERCA curve versus dobutamine curve, give you exactly the same increase in contractility. So the end effect in terms of improved function of the heart is the same, but all the dobutamine animals die because of arrhythmias, while most of the animals with SERCA survive. So you can see how a targeted approach really improves, gives you a very different effect. In addition, when we looked at the effect on the cardiac myocytes, which has this nice topology for the Z disc, and they line up nicely in a normal myocyte, in heart failure, that's architecture is completely disrupted. And you can see with restoration of SERCA2A, you basically have restoration of the topology. So clearly, what we're seeing by targeting this transporter is really a reverse remodeling, an improvement in multiple parameters, whereby we're sort of abrogating the adverse responses that cardiac injury induces. One of the issues with gene therapy in the cardiovascular system is you just have to understand compartments. And we're trying basically to put a gene in the cardiac myocytes, and there are about 10 to the 9 cells in the heart. So by doing direct injections, we would cause so much injury that it wouldn't be worthwhile doing the study. So we have to use the perfusion system, and you can do that surgically by open chest surgery, and that works. There have been multiple approaches using the coronary circulation, so you can navigate the veins, the coronary arteries. You can make some systems that are extracorporeal, so creating circuits outside the body and bringing them back into the heart. And we settled with a simple technique, and the reason we did it simply is because we needed penetrance in busy cath labs, where if you spent more than 2 to 3 hours, you're never going to get any time there. And it really included this sort of slow perfusion of AAV vectors down the coronary arteries. And by doing that, we're able to get quite a robust efficiency of about 60% of the myocytes infected with relatively low doses, so 10 to the 12 viral genomes. And you can see here where this is a clinical grade vector that we used. By increasing the dose, we're basically getting more and more exogenous protein and message formed in the ventricular walls. So prior to our studies in humans, we had multiple models of heart failures that we looked at. They're mitral regurgitation, porcine models of infarction, pacing model of heart failure in the sheep. So we tried over 200 animals, and obviously I'm not going to go into it, but they were clearly positive for us to basically take the next step. The immune response in terms of AAV gene therapy is, as we heard, is centered on 2 things as far as sort of we're concerned as you go into clinical trials. First, the antecedent neutralizing antibody. So when Dr. Burns asked us to raise our hands if we're exposed, yes, many people are exposed to 1 or 2 of the serotypes. And the second thing is the T-cell response. So when we took patients with heart failure early on to see how many of them had neutralizing antibodies, we actually found that about half of them had neutralizing antibodies of 1 to 4 or more, and these patients would need to be excluded from our trials because the neutralizing antibodies would wipe out the vector. And actually, I really didn't believe that because we were using 10 to the 12 particles, but this is what we saw. So if you took neutralizing antibodies from patients' serum and you incubate it with AAV vectors, you got complete wipeout once the serum had 1 to 2 or 1 to 4 neutralizing antibody. And in animals who have neutralizing antibodies, you get no expression. So this is a clear phenomena, and it has to be incorporated into the clinical trial. The second thing is much harder to predict, and because we can't predict it in animals, and that's the T-cell response. And the idea is that the capsids at high enough doses of vector are exposed to the circulation by MHC class I cells, and that would trigger a T-cell response. Now, we can't predict it. The only thing we know is that it is dose dependent. The higher the dose, the higher the likelihood of doing this, of getting this. And the only thing we could do is basically devise an ELISpot assay to basically monitor for this. So about 4 years ago, we started a phase 1 clinical trial, an open label sequential dose escalation trial in patients with severe heart failure. And we went 10 times, or the FDA asked us to go 10 times below the median dose of efficacy and 10 times higher. So we had 10 to the 11 and all the way to 10 to the 13. It was a very slow trial because we had to wait months between each dose and so on to make sure it was safe. But that phase 1 clinical trial gave us enough safety data and also even some biological signals that when we went ahead and started phase 2 trial, which at this point was double-blind, placebo-controlled. And the design was basically we had 3 doses of AAV vector and compared to placebo. The primary analysis was at 6 months, and the second analysis was at 12 months. The eligibility was based on usual criteria for severe heart failure. One added thing was that all patients had to have defibrillators internally, but since they had severe heart failure, at least in the US, they qualified to basically have either biventricular pacers or ICD implantation. And neutralizing antibodies of 1 to 2 or more were exclusion criteria. The procedure was quite simple. The patient would come in the morning of the procedure, basically undergo cardiac catheterization where the vector would be injected down the coronary artery. Everything was done with usual catheters. And the clinical events were driving our endpoints, basically. This is what we use for in heart failure, is worsening heart failure, hospitalizations, all-cause deaths, assist device placement, or transplantation. Patients were, this is sort of what the patients look like. This is the etiology of the heart failure, and both ischemic and non-ischemic. This was important for us because biologically, our hypothesis is that regardless of the etiology of heart failure, calcium cycling is abnormal, so you should not be sort of choosing the best scenario, which would be idiopathic normal coronaries, but really just take all comers with heart failure. These were maximally treated patients, so we can see the usual treatments for heart failure were maximally administered to these patients. And again, these were very sick patients, systolic volumes of 200, ejection fractions of 25, and BNP levels of close to 3,000. So in terms of heart failure patients, these were extremely sick patients. So this is a figure that basically shows what happened to each patient. So each line is a patient, and each of these circles represents an event, whether it's transplant, death, requirements of chronic inotrope, and you can see that the placebo group has a lot of these events, and then the low and mid-dose group do well for the first 6 months, and then catch up to the placebo group, and the high-dose group does quite well. And tabulated, this is what it looks like. The placebo group on top has continuous events happening. The first 6 months in the low mid-dose groups are good, and in the high-dose group, we see a very nice very few events. This was correlated with physiological data. This is days of CV hospitalizations, which are significantly lower in the high-dose group. BNP levels, which as the ventricle enlarges, get bigger and bigger. And again, you can see the dynamic state of the disease. So once you have that degree of heart failure, the disease is continuous and gets worse and worse. This was abrogated by the high dose. 6-minute walk test, which is in heart failure, is one of the tests we use to see how the patient is doing. That gets worse, and this was again reversed with circuit 2A. And end systolic volume, how big the ventricle is, continues to get worse in the placebo group, and that's again reversed in the high dose group. We now have data from up to about 3 years, and again you can see the placebo group, half of them has already died, so did the low dose group and mid dose group. Again, this is a disease that has a mortality of about 60 to 80% at 5 years, so we're not talking about sort of a chronic disease that peters on. And again, the high dose group, even though we have obviously more events at 3 years, continues to do much better than the placebo and the two mid-dose groups. We had no arrhythmias, new arrhythmias on study, so the patients who received the AAV circuit did not have an increase in arrhythmias, which was again one of the concerns because we're not infecting every cell, and there may be some issues of overexpressing circuit. The low and middle dose groups were quite sort of puzzling to us. One, because there seems to be a positive response in the first 6 months. And from the biopsy results, what we found was that there was really very little expression, exogenous expression, at the low and mid-dose group. The only expression we're able to see of the vector that we put in was at the high-dose group. So again, the higher the vector, the higher the expression. And so why did we see an improved response in the first 6 months with the low and mid-dose group? Well, when you inject the AV circuit down the coronary arteries, you're infecting endothelial cells. And what we were able to show was that, in fact, SERCA2a gene transfer in endothelial cells enhances eNOS, both in terms of its expression and in terms of its phosphorylation at serine 1177. And this translates into an improvement in coronary flow. So if you have improvement in coronary flow, which is nitric oxide mediated, your overall function will probably get better. Again, this is speculation, but that's really the only explanation we could count. The other question is, should higher doses of AAV circa be used? I mean, clearly, the higher the dose, the better the clinical effects that we saw. So this is where we are at 10 to the 13 viral genomes that we delivered at the high dose. Many trials that have gone above 10 to the 13, including the recent hemophilia trial, saw ELISpot positivity and had to use either steroids or something to rescue. And again, this is one of the issues that we have, is that in cardiac clinical trials, if you have inflammation within the myocardium, the patient may die, and that would be the end of the trial. So we kind of are stuck at 10 to the 13. I mean, we thought about it quite a bit, and I think we are sticking with 10 to the 13 as our high dose. The other issue is that half the patients with in CUPID had to be excluded because they had neutralizing antibodies. Now, if you have hundreds and thousands of patients with heart failure, that degree of heart failure, maybe that's not an issue. But one way to do it is to basically do plasmapheresis, and you can remove the neutralizing antibodies. A second way is to do designer AV vectors, as we heard, and you can make them specific to escape neutralizing antibody. That's exactly what we did. We used DNA shuffling and directed evolution by breaking down the AV serotypes and screening for libraries. And yes, we were able to fish out one AAV that had absolutely resistance to anything we threw at it, but of course, it was not cardiotropic. So there you go. So it's really not very useful, but after years of trying, we're able to get one characteristic change, but we lost a lot of other things by doing that. So in terms of the CUPID trial, so we are now, we basically, in November 2011, we got a fast track designation from the FDA. We are now in the middle, or not the middle, but the beginning of a phase 2/3 trial with 200 patients. This will be, actually CUPID-1 was the biggest gene therapy trial that's ever been done. This will be an order of magnitude beyond that in terms of a gene therapy trial. So we're going to enroll 200 patients in 50 international centers, where we're going to use the high dose of AV circa versus placebo, and the patients will be followed for 12 months. So 25 centers in the US and 25 centers in Europe, which are going to start enrolling these patients. Two other trials, one in London in patients with LVAD and SERCA2a gene transfer will hopefully start soon. And another trial at the Pitié-Salpêtrière in Paris is going to start in September, where they're going to be looking at LV function, specifically by multi-imaging modalities. And that's a separate trial. Other trials are being planned because of the biology of circuit 2A are in normal ejection fraction heart failure, primary pulmonary hypertension, and prevention of arrhythmias in patients with high susceptibility. And the French trial will also have a component where we will be basically collecting IPS and generating cardiomyocytes on our platform in New York and basically looking at some of the tests in vitro. So the journey really to go from the target identification, which was about 20, 25 years ago, all the way to the clinical trials was quite long. Vector development and testing of various vectors was one of them. Gene and cell delivery was another issue we had to basically develop ourselves. Molecular imaging were another part until we finally got here. So I just want to close with just just a few slides on, well, now that we're in clinical trials, do we stop here and just sort of say, okay, well, we'll just wait for a few years? One of the things that was very interesting to us, because we're targeting a cellular pathway, is what happens to the exogenous circuit that we put in a heart failure milieu. So if you look at circuit 2A in a normal pig heart, it basically has a PI that has an acidic distribution. In heart failure, it has a basic distribution. When you overexpress circuit 2A, what you see, unfortunately, is that new circuit is made, which is in a normal PI distribution, and then in the heart failure milieu, it basically gets post-translationally modified. And there are multiple reasons why you can get modification of a big protein in a heart failure milieu, and acetylation, methylation, nitration are all of them. And a few years ago, or about 4 years ago, we undertook an interactome strategy to look at what associates with circuit 2A. Many things do, and this is sort of a whole list of them that were validated after 4 years of work. And one of the things that was interesting to us that came out, which we didn't know about, was SUMO1, which was identified as a circuit 2A interacting protein. And SUMO1 regulates protein-protein interaction. Unlike ubiquitination, it doesn't only tag a protein for destruction, but basically it can induce conformational change, it can provide a binding site, or it can interfere with an interaction. And we basically found that it was decreased in the heart failure models that we have, including in humans. And we identified the two lysine sites that it was interacting with, and those were 480 and 585. You can see SUMOlation, here's circuit 2A. The SUMOlation basically adds these bands to circuit 2A, and when you modify the sites, you basically don't see any of those bands on top. So again, using a gene therapy approach, we were able to show that in multiple heart failure models, you can basically improve function. And this was very similar to when you improve function with circuit 2A. So here's LV dimension getting worse in our TAC model, aortic banding model. And with overexpression of circuit 2A or SUMO1 individually, you basically get improvement. This is the survival benefit of just overexpressing SUMO1. And basically, we're able to find that if you enhance SUMOlation, you basically, circuit 2A is stabilized, it increases ATPase activity, and improves calcium transients. And we have now an active program of SUMO1 activators, which is quite mature, having identified at least 2 large families that SUMOlate circuit 2A with small molecules, so we're quite excited about this. But one of the other things that we did was we basically created a vector where SUMO1 is incorporated, and the idea is to basically stabilize circuit 2A as it's being formed. And in a pig model of heart failure, we're able to show that this strategy gives you a synergistic effect in terms of circuit 2A function. So this is our program in gene therapy that has kind of emanated from our interest in SERCA2A. There are a couple of other trials that are upstream of SERCA2A that I don't have time to really go into. And with that, I'd like to thank you for your attention and acknowledge the work from many people in my lab for this effort. Thank you.
Thank you very much, Roger.